Hello, hello, madam. Can you hear me? Madam, I'm not able to hear.
How are you?
I'm fine, madam. How are you?
Fine. So nice to see you. Sorry to drag you early morning on a Sunday, in spite of you having a very busy schedule.
I have to go to hospital after this.
Okay.
Yeah.
What I'll do is I'll make you the co-host, so you can just share the screen and keep it ready.
Yeah.
And then we can start, right? Just wait for 5-10 minutes for some more people who are just joining us, right?
Yeah. Okay, madam.
I'm making the co-host.
Yeah.
You can now share your slides. I mean, you.
Yeah.
Excellent.
Can you see, madam, my presentation?
Yes, I can see. Fantastic. So we'll keep it at this, and then I'll just allow people to enter in.
Yeah, okay.
Just 5 more minutes, right?
So
How are you guys? All well?
All well, madam. How are you?
Bas, I'm doing well. Tell me one thing, since you're here. I'm doing a cervical spine for a schizophrenic patient tomorrow.
Okay.
Patient is going to be a cervical with lumbar. Patient is on valproic at night he takes, which I'm going to give him.
Okay.
And he's on Anaesthesia, which is that blonaseranin.
Okay.
That also I can continue, na?
What is the, is it an certain reuptake inhibitor? Or what is the group? Actually, I don't know the brand name.
Even, yeah, no, I looked it up. It doesn't say it is an SSRI. I did not. So that's why one is blonaseranin. I do not know what it is. It's called as ELICIA.
Okay, fine. It must be some newer agent. Most of the new agents are fine to be continued.
Yeah.
That is the only thing.
It's used to treat manic episodes of schizophrenia disorder.
Yeah.
That's what it says. So
how practically it will affect is if you don't give the medicine on the day of surgery, he might have post-operative delirium, and episode of schizophrenia can come in the post-operative period, which will confuse us with a delayed recovery, bar delirium, bar cognitive dysfunction kind of situation you can look in. So at the same time, giving the tablet on the day of surgery can lead to some hemodynamic changes during the surgery. So since you're a cardiac anesthetist, you should be okay with that.
Yeah.
So if you're okay with it.
Though he does not have any comorbidities, but I do plan to put in an outline in this patient for the same reason, for the hemodynamics. That's the only reason.
All these cardiac abnormalities that the psychotic
have. Yeah.
All these things were in the era where the modern monitoring system was not there. Cardiac output monitors. When you have all these things, you shouldn't be worried about the slight hemodynamic changes that these drugs cause.
Yes, yes.
So, but you should be worried about the post-operative cognitive dysfunction if it really happens, because that's not in our control.
So, do you think I should avoid this synthetic, like tramadol? I should avoid as much as possible.
But tramadol is a very benign drug, but it doesn't affect that much into the central nervous system.
Okay.
So, you just have to keep the patient in the right depth. Actually, that's what I'm telling you. This patient,
of course.
What happens is this psychological disorder patients are more prone for going in. They're very sensitive for anesthetics. So without our knowledge, you'll be overdosing them most of the times.
Correct.
If you try to keep them in the right plane, they'll actually wake up brighter.
Yeah.
So, that is one thing. Post-operative, if you feel the delirium is there, there are different ways to tackle that. One is giving metazolam. Some you can run serenade. Some people give. Some people run Dexam low dose for some time. But some kind of calming agent is required in the post-operative.
The other thing I wanted to ask you: when you're doing your cervical spines and your patients are prone, in a prone because of the posterior approach, do you put your PIS monitors even then?
Yeah, in our center, all the cervical cases will be done on Sugita frame. Sugita frame is kind of frame where you fix the head on a 4-pin system.
Achha.
So 4 pins will be fixed here. 2 will go above here, 2 will go on the forehead in the
Right.
So you will have enough space here to attach this.
Correct.
So that is the reason I'm able to do. And
but what happens in prone? Do you think you get adequate this thing, because it is when you're using that dress for the orthopedic people? Do you think
sometimes after there is one way of fixing Ioban, Ioban, the yellowish color.
Yeah, the
So,
beta.
yeah, that one. That
Correct.
if you fix,
Ah,
most of the times it won't come, but if it might get loosened up also after 1-2 hours.
correct.
Possibility is there. In my center, I'm using EEG monitoring there directly,
Ah,
which is connected to patient by a corkscrew electrode. So
ah,
screw we turn and twist into the skull. So it's a little invasive, but
right.
the impedance comes to near zero, and there is no way it will come out during the surgery.
Hmm. We get a
So
lot of evidence with BIS because we are using cautery, we are using
yeah, but that is the problem with this.
on. Yeah.
And somehow the company guys, I don't know why they're not solving that problem.
Hmm.
I think that is a serious problem with this. That is why nowadays I'm liking the patient state index more than this. So we're planning to buy that, and I did a demo also for 2 weeks. Patient state index is superior, I feel. So. Even in the recent workshop also told the same thing.
Okay.
Patient state that SedLine monitor they call in Massimo, it will come.
Yes.
So that's better.
That's headline.
It's better, and the artifacts are lesser in it. All those things I'll cover today.
But till that we don't have one, how best can we utilize BIS?
How best is
The IOBAN technique that you said?
IOBAN technique is first luck should be there. If your luck is good, you'll get BIS for the whole case. And the second thing is IOBAN technique. You have to clean the area properly. So some actually prefer to clean it with sterilium or spirit. Nice.
That I always do it. Yeah.
Dry it and then attach.
Yeah.
And put IOBAN on top of that.
Correct.
So that much only you can do, madam. After that, see, after connecting and drying the patient, and if it loosening and if the signal quality is deteriorating, you cannot do much about it.
Right.
You can't keep going down and removing the drape. And
you can't.
It becomes very difficult actually, because you need to focus on multiple things.
And what about, do you get in these schizophrenic patients more of burst suppressions? Have you noticed that? Or
yes, I noticed that. And schizophrenics and dementic patients and Alzheimer's patients, Parkinson's patients, and patients of senile dementia and aged patients more than 90 years. 80 is okay, above 90. And little dull people, when you see them, you'll feel frail, frail and dull people. Correct. Without our knowledge, even with 0.6, 0.7 Mac of CO and S, they go into burst suppression.
So you feel that, I'm sorry, numerically 40 to 60 that you keep the range, you like to keep it air more above 50 on these patients to avoid A, burst suppression, and B, to keep them.
Basically, if you're following BIS, then it's better to keep between 40 to 50. 50 plus would be little. Yeah. The only problem is they won't be aware, but for every stimulus, there will be a response. Okay. Keep that light. But it is impossible for them to have awareness during, because they're hardly aware even in the normal situations. So they won't be aware. But the thing is, the stimulus will keep coming, because we maintain mostly on no relaxant technique in all the cases. Yes. Only for lumbar spine cases where monitoring is not there, we run relaxant cisplatin infusion. And even intracranial cases, most of the patients are being monitored now. So for me, actually 70 to 80% of the cases are run without relaxant. Right. So depth is a very important thing for me. Since we don't have remifentanil in India, the anesthetist skill matters actually. If you have remifentanil, the anesthetist skill, I don't call it, it doesn't matter, but even with lesser skill, you can manage. Remifentanil can take care of the patient. I went to Japan, I saw how the juniors are managing the most complex cases there with remifentanil. It's very easy actually to manage with remifentanil. Okay, I think most of our students have joined, and if you're okay, may I start? And Dr. Dheeraj, with your permission, may I record this meeting? And yeah, please record, madam. I am open for recording. Most of my almost every alternate day, my recordings will be coming in YouTube. Right. Okay. So good morning, everybody, and I welcome you all again to the DNB Clinical Meeting Series. We had a lovely talk yesterday with our HOD, Dr. Alka Halbe, and today we are continuing with our neuro series. And today we have none other than a very dynamic young neuroanesthesiologist from Sakra World Hospital, Dr. Dheeraj Masapu. I had the good fortune of being with him as a co for
2 workshops that we conducted. He's a very experienced consultant, and he has, with a history of working in the hospital and the healthcare industry. He's very skilled in operation management, operation excellence, and he's also a Six Sigma management and healthcare management. And he is very focused in neuroanesthesiology. He has passed from National Institute of Mental Health and Neurosciences with an experience in intraoperative neuromonitoring techniques, in which he's got a forte, and for safety of complex neurosurgical cases. And he's interested in neurointensive care management as well. He has, of course, published a lot of articles in neuroanesthesiology and neurointensive, and neuromonitoring is the area which he really works on for critical patients, and using different neurophysiological techniques. I was really enamored by the way he works when I interacted with him, when I had the good fortune of meeting him at 2 of the workshops. He also has a very, 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 very nice YouTube video, which is going to be very helpful for all the residents. And I think I had shared the link with my residents as well, which I will reshare again if other people want to do in the chat box. With this, I'll give the forte to Dr. Dheeraj Masapu to take over. And if there are any questions, kindly put them in the chat box, and we shall surely answer them. And with this, I also welcome a very senior neuroanesthesiologist who's been our mentor and a patron for this series, Dr. Nayak, who's there with us today. And Madam has been kind of a pioneer in neurosurgeries in Bombay, and she has been the ex-HOD at KM, and then she was at Nanavati Hospital, and now she is enjoying her retirement, but she's continuing her education and imparting education to our students and giving excellent tips. Thank you, Madam Nayak, for joining us. And with this,
thank you, Kalpana. Thank you, Kalpana.
You're welcome, ma'am. With this, I welcome Dr. Dheeraj Masapu, and the platform is yours, Dr. Dheeraj.
Thank you very much, madam. Actually, madam has given a very good intro about me, but I would like to give a self-intro about me. So I did my MD in a private medical college in Andhra Pradesh. After that, I was doing private practice there in Andhra Pradesh, and then I started doing some neuro cases there. So then I got interested in neuroanesthesia. And thank you, Dr. Nayak, madam, for attending this particular presentation. So after working there, I realized I need to upskill my game in neuroanesthesia. Then I did my DM in neuroanesthesia and NIMHANS in 2013. So since 7 years, I've been working as a consultant and in charge of neuroanesthesia in Sakaraval Hospital, Bangalore. So this is a brief intro. And my interest. So my basic interest is in neuroanesthesia and intraoperative neuromonitoring for brain and spine surgeries. So that is a brief intro. And so can you see the screen, madam? Kalpana, madam?
Yes, we can see the screen. I just got a message from somebody who wants the volume to be a little more louder if it's convenient.
Yeah, that is the problem. My voice is not so loud actually. Every time I use a speaker mic when I do YouTube videos. Now, 1 second. So is the volume okay now?
Yeah, I think it's good enough for me.
Okay, fine.
I can hear him very well.
Yeah.
Yeah. Okay. Thank you, Mike. So let us. Whenever I go to a conference or any presentation for depth of anesthesia, they give me 8 minutes, 9 minutes, and all. So what happens is I rush up the presentation. So today, madam has given me more time, I think. Right now, madam?
It's all the time is yours.
So I would love to go a little slower with the presentation because as an anesthesiologist, I think depth of anesthesia is a very important topic. Everyone should have a clear understanding about that. So whatever I understood in this decade, I'll just tell you. So there are certain situations when I give anesthesia to patients, I get really confused. So this is one of the cases. So what happened is patient of cervical IDM. IDM is intradural and extramedullary tumor. You can, I'll just try to zoom in once and show you. So this is what the IDM looks like. So this is a particular case where laminectomy will be done, and along with that, the tumor excision also will be done. So during the process, we monitor something called as motor evoked potentials and somatosensory evoked potentials. For the potentials to come during the surgery, so what usually happens is any agent which acts on the GABA system like propofol or sevoflurane or any agent will suppress the motor evoked potentials. It's very tough to get a baseline motor evoked potentials in these cases. It's very easy to get in a normal patient, but in this particular cases, with my experience, I found that it's very tough to get. So why I'm telling this is, so in these kind of cases, how do we manage the patient? So if I run certain drugs, it will get inhibited. So then I came to know some anesthetists are using dexmedetomidine and ketamine combination for some cases, small cases. I thought, why not use this technique in that particular patient? So when I used only dexmed and ketamine in that particular patient, the thought process of mine was going like this. So will the patient move during the surgery? Because we don't use muscle relaxants when we are doing cases for neuro monitoring. So since I'm not using a base anesthetic like propofol, which I know that surely will inhibit the movement. Using this kind of combination, will the patient move? Will the patient be aware during the procedure? And what happens to IO, whether it will come or not. So theoretically, dexmed and ketamine should give a good motor evoked potential response. So, but practically, will it translate or not? And post-operative recovery, since the surgery goes for 8 to 10 hours usually. So post-operative recovery, it's my responsibility to wake up the patient maybe within half an hour at least, or maximum 1 hour. So using such kind of a combination where dexam is known to have accumulation in the body, and ketamine is also known to have some kind of accumulation for long duration. What is going to happen? So these are the kind of questions which runs in a consultant's mind. Okay. So when I monitored EEG, I usually monitor EEG for all the patients. In this presentation, I would like to explain how I actually use. So in this particular situation, the EEG was showing that patient is in mild sedation. Then I got really worried if the patient is actually hearing what we are talking and is he having awareness. So this is one scenario. So what happened in this scenario, I'll tell in the end of the presentation. Because if I tell you now, you will not understand. Once I explain you how the EEG works, then you'll understand actually how the case went. And another case is a scoliosis case. It's a very regular scoliosis, will be done in many hospitals on a daily basis. So just check out this video, what happened in this particular case. So here, if you have seen scoliosis, you have seen something called as burst suppression. And the EEG is called burst suppression. And I remember the TCI is going at 4.5, which is a very good dose of TCI propofol. But despite of that, this patient is in a depth of burst suppression, which is very deep. So what we did in this particular case in scoliosis correction, I'll tell in the end of the presentation. Once I explain the basics. Another case I would like you to see is a spine surgery case. A very high dose of TCI is going on, 5.5, and dexmed also is running. And we don't use inhalation agents, I told you, because we monitor the motor evoked potentials and somatosensory evoked potentials. So these are the motor evoked potentials. And the EEG of the patient is fine. So this is how the EEG looks like. Right now, you'll not understand, but at the end of presentation, you'll know how to interpret EEG. This is somatosensory evoked potential waveform. It's called SSCP waveform. The spectral frequency this is a spectral frequency parameter. I'll explain you. So in this patient, the parameter is showing that the patient is little lighter. Despite of using high propofol, TCI, and higher and dexam combination, the patient is not deep enough since the patient is young. So these are three different scenarios. In the end of the presentation, I'll tell you how I tackled each different scenario. Before that, we'll go into the presentation to understand actually how the depth of anesthesia monitoring is done and what are the different ways to monitor and how the EEG system works. Madam, can you hear me now, madam? Till now?
Yeah, very well. Just
Okay.
if you could, sometimes when you go away from the mic or what your source is, I do get a lot of changes.
Okay, volume is coming down. Got it.
Yeah, yeah,
Now it is fine. Okay, fine.
yeah.
So most of us know propofol is what we use in neuroanesthesia, and there are two situations which can happen. One is if you overdose the patient, there is a possibility of hemodynamic collapse. So what happens is sometimes you see when you run TCI, BP will be low. Some people start phenylephrine infusion, and some people start NORAD infusion. So all those things might be prevented if you actually keep the patient in the right plane. And if you underdose the patient, worrying about the hemodynamic collapse, then there is a possibility of awareness. So how to identify the sweet spot? And I always tell everyone in the presentation, BIS saves lives. So the reason is, see, nowadays what happens is the age group of the patients is, the mean age group of the patients coming for surgery is increasing, particularly neuroanesthesia and spine surgeries. Most of them are 70 plus, 80 plus, sometimes 90 plus people are coming. So when this kind of people are coming, so if you might be thinking that you're running a normal anesthesia and a normal TC, a normal sevoflurane, but you might be overdosing them. And overdose leads to hemodynamic collapse. You end up giving more fluids and inotropes, and there is a possibility of postoperative volume overload or perioperative MI risk is there. So keeping them in the right plane actually saves lives. So I have practically seen many people where without my knowledge, I was overdosing them, and I corrected the path by using this. So it is not, depth of anesthesia is not a new thing that we are doing now. Since ages, people, the anesthetists have been doing it by looking at the physiological responses like hypertension, tachycardia. When surgery is happening, if you get tachycardia and hypertension, that means that one of the reasons might be a lighter plane of anesthesia. So this is how it is being done. And now we have advanced technology, so why not use it? And this is Guidel staging, and stage 1, stage 2, stage 3. So most of the times our patients will be in stage 3, plane 2 or 3. So that is the plane we need to target. So that will exactly correspond with a BIS of 40 to 60. So let us try understanding what an EEG is. If you understand this properly, you'll understand how BIS works, how entropy works, and how patient state index works. So actually, the EEG is an electrical signal which is originated from a cell called as a pyramidal neuron in the brain, which is located in the cortex of your brain here. So EEG signal, the main difference between an EEG signal and ECG signal is ECG signal will have a rhythm. So when you see an ECG, there is a rhythm. There is a QRS complex and a T wave and a P wave, and it comes consistently without any change in that. If it is changing, we are worried. So, but EEG is not like that. EEG doesn't have a rhythm like an ECG. It comes very chaotic and haphazard. So what scientists try to do is they try to find meaning and sense to the haphazard rhythm of the EEG. So in the process, they're inventing all the gadgets. So that is how the scientists are when they're trying to find the rhythm. So when they found the rhythm by a technology called as the spectral analysis or spectrograms, they divided the EEG into 4 different patterns. This we read for our entrance exams. Delta is when the patient is beta is when patient is awake, the frequency of 12 to 30 hertz. Alpha is when you close your eyes and when you're awake and just close your eyes, you get an alpha rhythm. That's 8 to 12 hertz. And theta is a mild form of a sleep, and delta is a deeper sleep. So this is how the EEG is differentiated. But in reality, what happens is what we were told, I don't call it totally wrong, but it is partially wrong. Because what happens is at one point of time, all the waves coexist. When you're awake, you'll have all forms of waves. All these 4 waves will be there. Predominantly alpha, beta will be there, but you'll have some element of alpha, theta, and delta. Under anesthesia, predominantly it will be delta, but there will be some element of other waves also. So all the waves coexist. That is how brain works. So this is an image taken from a very famous article published by a famous man, and EEG changes in different phases of anesthesia. If you can see I'll try to use laser point. So this particular wave here, This particular pattern is seen when you're awake. Here, the pattern is straight. It is straight and a faster frequency, if you see, and a smaller wave. I'll tell in a very simple way so that everyone understands. And when you close your eyes, the beta oscillations start. Okay, so directly we'll go to this part of the wave. When you give anesthesia, what happens is the wave which is straight is converted to a wavy pattern. So this is our target under anesthesia. And if you keep the patient very deep, this pattern will start. Here you can see a suppression followed by a burst. This is called burst suppression. We don't want this pattern. We want this pattern. So let us try and zoom in and understand what this pattern is and why are we targeting this. So to understand this, a very interesting concept I would like to tell. So what happens is, when you're awake, and if we do EEG of your particular brain, then we see different types of waves coming from different parts of your brain actually. Not every part of your brain will be same. Okay, so that is how brain is different from other organs. So in awake state, what scientists observed is the posterior part of your head, that is your occipital area, will be having the alpha pattern predominantly. And when you give anesthesia, what happened is, what they noticed is, the alpha waves will shift from back to the front. This is called as anteriorization of alpha. Very simple concept. The alpha waves which are located in the back, they come to front. So you'll see more predominantly alpha waves in the anterior part of your head. Let's see the next. So then what happens is, so why are you telling us this concept? If you ask me, why I'm telling is, so since you place your BIS here, you are actually trying to see how much of alpha pattern is there. Okay. And since I showed you in the particular image, there is a wavy pattern. If you zoom in, this is the EEG and this is the wavy pattern. If I zoomed over two particular waves, each wave, you can see the yellow line, each wave is called delta. So the delta, what you actually study is not a single wave. It's a culmination of multiple waves. Multiple alpha waves form one delta. So this is called as background activity by neurologists. And we need to get a background delta activity. And on top of delta activity, you will have the alpha pattern. So this alpha has come there, since I told you from posterior to anterior, the alpha shifts there. So alpha on top of delta is the target for us during anesthesia. I think, I hope everyone understood how so this is the basis for all your algorithms of BIS and entropy and patient state index to derive a number. So this is the basis behind all the advanced monitors. So they don't tell you the algorithms because BIS has not released their algorithm. They keep it secret because they will have a patent. So when they make an algorithm. So how the algorithms work is this. So when you understand. So anything, what I believe is when you understand any concept from the roots, then it will be saved in your permanent memory. Anything you understand superficial, you'll usually forget within 24 hours. So now I think this concept I try to explain you from the roots, you might remember this for a longer duration. So this is how your BIS monitor looks like. That was my monitor. Because I'm a neuroanesthetist and I have advanced monitor, I can derive the EEG. But BIS is a common thing. Everyone can afford a BIS. So this is how BIS looks like. You can see. Madam, you are able to see my laser pointer, no? There.
Yes, very well.
Yeah. So this is what I was trying to show, a wavy pattern under anesthesia. If you can achieve this target, your BIS number you don't have to see actually. BIS will be patient will be in a right plane. So on top of each wave, there are multiple small waves. It's called superimposed pattern. It should be alpha. So alpha on top of delta is the pattern that we desire to achieve with a good depth of anesthesia. So then you're sure that you're not overdosing him. You're sure that he is not aware. And I am pretty sure that he won't move without relaxant during a no relaxant technique. So for me personally, I have another parameter which you people won't be having. It's called spectral edge frequency. It's more sensitive and specific because BIS is prone to have some artifacts. We were discussing before you guys joined in. BIS is prone to have catch and artifacts. The sensors get loosened sometimes. But we use corkscrew electrodes and we get spectral edge frequency. We try to keep this particular number you're seeing between 12 to 15. So this is derived from something called as digital spectral array, which I'll be explaining later on. It is also a simple concept. You'll understand it. I'll explain a simple way. So this number is very important for us. So this is called as it's called a spectral analysis. So like ECG, like an ECG to read, it is very simple. But EEG, since we cannot read, the spectrogram has been invented. Spectrogram, what it does is it tries to differentiate different frequencies of the EEG. And for a simple person to understand, they color code it. So they color code it in such a way, you can see here. This is color coding. And then what DSA does is, it gives red color where maximum number of frequencies are located. For example, if the patient is having predominant delta activity, then the red color will be located between 0 to 5. If the patient is having a superimposed alpha activity, you will have another layer of red color at between 8 to 12 mark. So by looking at DSA, roughly I can tell that his frequencies are distributed in such a way, some frequencies are here and some are located here. So he is having an alpha and delta. I think this patient is in a good depth. So, and this DSA is being given by the Massimo monitor, actually. Patient state index and Massimo, I'll show you in the end. So once you see this, so why can't we see the number? Why should we go into such a depth, if you ask me? Number can mislead you sometimes. So basically, a surgeon who's operating is not bothered whether you're seeing a number or whether you're seeing this. He doesn't want the patient to move during surgery, and he doesn't want the patient to have a delayed recovery. To achieve the target, you need to have your own plan. So this is a way you can be more specific in identifying. So these are the every anesthetic will have a different kind of a DSA. I'll try to explain here. This what I explained is propofol till now. If you can look in this particular diagram, so when this is the whole phase of anesthesia, when you induce the patient with propofol, most of the frequencies are in delta range. Patient will be very deep. Once you start the maintenance TCI, then the split happens. There will be alpha on top of delta. So there will be some frequencies in alpha and a superimposed, alpha will be superimposed on delta. When you start recovering the patient again, delta will disappear. This is the plane where patient recovers. So the whole, by looking at DSA, you can tell which phase of anesthesia you're there. And sevoflurane has another kind of a pattern. So when scientists experimented with CO and DSAs, what they observed is you will have a theta on delta pattern. You will not have an alpha on delta like propofol. As I told you, the shift happens like this. That is for propofol. But sevoflurane, it's a different kind of shift. There will be a theta shift here. So theta waves will come anteriorly when you measure. So theta on top of delta will be seen. So you'll have the two bands are very close here. If you observe, the difference is more here. Ketamine has a unique pattern. It will have delta, but the superimposed waves are in the range of beta, or sometimes they're called as gamma waves, even more light and faster than alpha waves. So that is why ketamine causes hallucinations and delirium and all that. That is all because of the glutamate excited opposite. So glutamate excitation leads to the gamma waves. So only till beta, above beta is called gamma. So gamma and delta pattern is seen in ketamine. By looking at this, I can tell that you are given a dose of ketamine to the patient. By looking at the DSA, I can tell what anesthetic you are using actually. So every anesthetic has a different type of DSA. This is called as EEG signatures. So I explained about propofol, I explained about CO, I explained about ketamine. I didn't tell you anything about dexam. Dexam wave is even more unique. So dexam, what it does is, it bears the EEG. That means it strips off the superimposed pattern on top of the delta. So delta looks bare. So here only delta is seen. The superimposed waves are removed by dexam. So when you're using dexam, only the delta pattern is seen. There is no superimposed frequencies here in the DSA. So if you're running dexam for the patient in higher doses, more than 1 mic per kg per hour, if you use this kind of pattern might come. So when you're using in that kind of levels, I can tell that dexam is being run. So I think, I hope you guys understood till now. Till now, if you understood, then the complex and difficult part of the presentation is over. Madam, can you hear me, madam?
Yes, very well, very well.
Any doubts you have, madam? After this, it is simple actually presentation. Till
Yeah.
now, actually, is
I don't see any questions in the chat box, so I'm sure
because
it's very
it is a tough topic, complex area. No, nobody can ask questions here. But I hope at least 50% if you can understand in what I have told you, you actually have understood the significant part of EEG. So let's go into the practical aspects now about BIS. So BIS is FDA approved technology. BIS is nothing but an algorithm. So what they did is they use something called as a time domain and frequency domain and bispectral analysis. And they use all the analysis and then they try to derive a number from the particular waveform. So since I already told you, so what BIS does is if the patient is having complete beta activity, it will take it as 100, saying the patient is awake. If the patient is having a flat line in the EEG, below burst suppression, there is something called as a flat EEG, which is usually seen in very deep anesthesia or if the patient is brain dead. So that kind of EEG is considered as 0, which you normally don't see under anesthesia. And if the patient is in the plane, which I explained till now, a wavy pattern on alpha on top of delta, it will consider that as 40 to 60. That is a recommended range for general anesthesia. Anything lighter than that, it will call it 60 to 90. So your target is to keep the patient between 40 to 60. That is the recommended range for general anesthesia. So BIS comes with, BIS will have BIS electrode, which goes on top of your head. So this is how BIS electrode is attached. It will have 4 sensors on top of that. Number 1, number 2, number 3, number 4. So number 1 is ground electrode. Number 2 and 4 are reference. And number 3, where you're seeing on the temporal side, is the active electrode. So the active electrode actually catches the EEG and rest are for references. Any electrophysiological wave needs to be compared and with a reference wave. Only then you get a number. So that is why 2 and 4 are reference. 1 is a ground. Ground electrode is there to silence the artifacts. Without our knowledge, there are a lot of electrical artifacts in the operating room, which are generated by table, by ground, and baseline electrical activity will be going on. All those are neutralized by the ground electrode. So you have to clean the area on the forehead with alcohol or spirit or sterilium and then let it dry and then apply it in the way which I have shown here. So after applying properly, and then you have to do something called a sensor check. So you have to make sure 4 ticks come. All 4 sensors are in contact. So what do you mean by contact? Contact is that impedance is in a reasonable range. Up to 0 to 5 kilo ohms is the impedance that we target. It's a reasonable level. Above 5 kilo ohms, it's not acceptable. I mean, the value that you get is not real. It's an artifact. So between 0 to 5 kilo ohms, if you have, then a green tick will come. So once 4 green ticks are there, then you can rely on the number that you're seeing. So once you see the number, don't directly go to the number of BIS. That is the wrong thing that many people do. First, you need to see something else. So first, you need to see something called as the signal quality index. In the image, if you're seeing, this is the signal quality index. If the sensors are properly connected, if the impedance is low, then the signal quality index will be above 90. So our target minimum 80 should be there. Above 90, you get a very reliable number. And then you need to see something called as an EMG ratio. So I will talk about the EMG in later part. So normally what happens is a frontalis muscle is there. Frontalis muscle generates a strong electrical activity is called as electromyography. So I'll tell you the frequency ranges in later part of presentation. This can actually contaminate your BIS number. So if the EMG is higher, normal range is between 25 to 35. So this number 29 is acceptable. If it is above 35, then the EMG is actually contaminating your BIS. The value of BIS that you're seeing is falsely high. You can't rely on that particular number. And then you have to see something called as a suppression ratio. It should be 0. Suppression ratio is a complex understanding. Let us not touch that now. And after confirming all these things, then you go and see the number. So once all these things are in reasonable ranges, then the number is also reliable. Otherwise, number is wrong. Okay, this is a correct number, 49. The patient is in correct depth. Okay, hope you're understanding what I'm trying to tell. And these are another 2 scenarios where this patient is too deep. 27 is very deep. and the signal quality index is 97. So I can rely on this number. I have to decrease the depth here. And these are the same patients. So I had to decrease the depth here, and this is the correct plane, where 40 to 60 is my target. So coming to EMG, as I told you, you have to understand this concept. The BIS algorithm works in such a way, it interprets any waves within the frequency of 0 to 47 Hertz. Unfortunately, between 30 to 47 Hertz, the EMG activity also will be there. So in this particular spot, both EEG and EMG both are coexisting. So that is the reason EMG can produce an artifact if the value is more than 35. So whenever EEG number on the monitor is more than 35, it can lead to falsely higher BIS. So that is the time. So if you ask me a question, can we rely on the number? What should we do at that point? What should we do at that point of time? The answer is, why is the EMG artifact coming? Because frontalis muscle is working. Give muscle relaxant or give painkillers, fentanyl. Sometimes it's nociception also can trigger EMG activity. There are studies. So give a dose of fentanyl and muscle relaxant, suppress the EMG, and then see the number. That is how you need to solve the problem. So this is a case where you see suddenly there is a rise in the BIS. You might think that if you don't understand the EMG, you might think patient is in a lighter plane, but actually the EMG activity also has gone up during that time. So that is the reason for elevation of the BIS value. So it's a false elevated BIS. You don't have to treat it, but I treat that because even EMG is not acceptable for me because my patients are not on muscle relaxants. So I want the EMG to be silent. I'll give a dose of fentanyl or morphine or muscle relaxant I can't use, so I take the patient deeper with opioids during that point of time or increase the TCI from at least a 0.5 micrograms per ml from the running dose. So this is another case where EMG artifacts, you can see spikes in between. These are called EMG bursts which happen and falsely they elevate the BIS. So this is a case where patient is in a lighter plane. I was running a TCI of 4.2. I think most of you are well versed with TCI pumps. If you're not, then you have to attend our workshops which we'll be conducting. So TCI is 4.2 is a good dose. And this particular patient is not in a good depth. He's lighter. So what I'm trying to tell is there are situations where you feel that patient is in the right depth, but patient might be lighter. But what I'm trying to tell, 63 doesn't mean that he'll be aware. For awareness, it should be above 80. 63 means that patient is not aware, but at the same time, he might respond to a stimuli. For example, he might get a tachycardia. Here you're seeing heart rate of 101. Maybe that 101 is secondary to the depth. So the stimulus response will be there, but he might not be aware. Even that is not acceptable for us. So extubation also, if you connect this, why not extubate with this? Don't remove when the surgery is over. You wait for the patient to recover on top of this. That is what is the ideal way to do. Here is the one case where extubation time, patient is 68. So then it will wait. I usually use no touch extubation. So I don't stimulate the patient. I just cut off everything, wait for the patient to get up. Do one suction in deeper plane. After that, don't do suction also. I hate it when somebody does suction multiple times and stimulate the patient and wake them up in delirious plane. So that doesn't work out in neuro anesthesia. In neuro, the extubation should be in a fully awake state. So we wait for the patient to get up. 68 to 75. 75, then I'll be like, okay, some more time we'll wait. Then 80. 80, I know, I'm sure that this patient will be awake, little drowsy. That is enough for me. And you can see 80 is the BIS value here. At the same time, so we're trying to wake the patient up. He'll open so smooth. So that is how it goes. So if you actually wake up the patient in the right plane, patients don't bug, the hemodynamics won't shoot up, tachycardia won't come. So it's not about using esmolol or labetalol or NTG during extubation. It's all about extubating in the right plane. For that, patience is required, and you have to deal with the surgeons because they will always push for an early extubation. You have to deal with them, and you don't try to explain them because most of them, these are very finer aspects. Many people don't understand. You just have to delay extubation for some time to get this plane. So limitations of this. So basically, this has certain limitations which actually covered till now, the EMG and all those things. So some anesthetics like ketamine and nitrous oxide and other agents, when you're using, they can confound with the values. As I explained before, when you give ketamine, it releases, it causes glutamate excitation, and there is a possibility that the this value may be falsely held. When you use Dexam on the other side, there is a possibility that patient might look more deeper than the present state of anesthetics. So these kind of problems you can have when you do polypharmacy. But what I observed is all those things happen when the dose of that particular drug is in a very higher level. For example, when you're adding dexmedetomidine, which is very commonly done, when you use it in the range of 0.2 to 0.5 max per kg per hour, nothing is going to happen. EEG is just coming just like normal. When you're crossing a dose of 1 mic per kg per hour, only then we are seeing these changes. So whichever anesthetic is actually controlling the patient will have the predominant EEG signature. So if propofol is the predominant agent which is controlling the patient at that point of time, then the EEG will exactly look like how propofol's EEG will be. So that's how brain works. Children, can you use? Some people ask me in conferences. See, this less than 6 months of age is not validated, but I try to use in some people just to understand. But based on that, I never interpret anything, but try to understand how this comes. It is just coming normal. I observed that even in children, we can get a very good EEG signal and titrate the anesthetics. Maybe in future, we might start using in younger kids. And hypothermia is a very dangerous thing for anesthesia and this also. In hypothermia, what happens is the EEG will become, in hypothermia, falsely, the EEG will show that patient is in a deeper plane. So then what happens is you will actually cut down the anesthetics and patient might be aware or bug during surgery. So it's always advisable, since we keep the patients in normothermic range. Nowadays, we are not seeing this problem. This used to happen before. Now, everyone is very vigilant about temperature management. We don't allow below 35.5. And in neuroanesthesia, when you're doing motor evoked potential monitoring, if temperature falls less than 35, the neurophysiologist will never agree. He will not interpret the MEPs till the temperature picks up. So you have an obligation and you are pressurized to keep the temperature normal. Neurological disorders. In the before the presentation, we are discussing with Kalpana madam, and any neurological disease, let it be schizophrenia, Parkinson's, dementia, or Alzheimer's, or any kind of post-stroke patients, post-head injury patients, post-brain tumor surgery patients who are little dull and drowsy, and those kind of people, they will have a lower baseline BIS. So those kind of people, you actually have to cut down the anesthetics. You have to decrease the anesthetics. So BIS is very important in these kind of cases. So if you don't use BIS, you'll overdose them. So when you overdose them, they'll get hypertension and also postoperative cognitive dysfunction. Very common in these patients. I have seen some people going into postoperative cognitive dysfunction for days. You might be thinking if it is for 30 minutes, for 45 minutes, 1 hour, you can deal with that with some sedation. It is not so. Some people go into cognitive dysfunction for days and some people don't come out. So you need to be extremely careful when you're anesthetizing neurological disorder patients. This is one of my study where we observed BIS in head injury patients when they're coming for decompressive craniotomy. What we observed is patients where the BIS is, patients where the head injury is severe and the baseline GCS is low, the BIS is also significantly low. So they actually didn't require much anesthetics for induction as well as maintenance. So all these days we were trying to overdose them with the regular doses. So they require a very less anesthetic, and so our priority should be to maintain the hemodynamics in head injury patients rather than keeping the correct plane of anesthetics. So that is the take-home point that we got from this particular study. This is another study of mine which got published recently on BIS. So we do something called as a D-wave recording during spinal cord tumor surgeries, and when we record that particular wave, BIS was falsely showing a higher level. And then we came to know that D-wave stimulation is actually causing an artifact in the BIS, and we published that. So we need to be careful. There are multiple artifacts around you. That's what I'm trying to tell. This another artifact is published by my friend Rithiman, and I am also part of it. And here what happened is during facial nerve monitoring, which we very commonly do for CP angle surgeries and trigeminal schwannomas, whenever they're stimulating the facial nerve, the BIS values are going up falsely. So if you have understanding about the facial nerve, then you'll interpret. If you don't understand that, then you'll be thinking that patient is lighter and you'll be giving some propofol and opioids and other relaxants to the patient. Hope you're understanding what I'm trying to tell. So even cardiac artifacts are seen, ECG artifacts are seen, and electrical cautery artifacts are seen. When cautery is done, it produces a very strong artifact on breast. So I usually don't talk about much evidence. I'll just show my studies and my practical experience with in any presentation. But this one study I would like to show you. So in 2007, Carlos et al. Found that the extubation and discharge from the PACU, the post-operative care unit, is significantly reduced when used BIS monitoring. It makes sense because when you keep the patient in a correct plane, the delirium will be lesser, the recovery will be faster. So that is how you need to use. Economic benefit is also there. When you try to give anesthetics in optimal way, you actually cut down the costs. You don't overdose the patient. Economical benefit is also there. So this is a final recommendation, sir. BIS is not only for awareness. What we actually think, BIS is to improve the anesthetic delivery, so and post-operative recovery. So that is the main advantage of BIS, which I'm emphasizing in this presentation. So NICE guidelines recommend BIS monitoring when you're using TIVA. Okay, so that is the recommendation from NICE. So this is about BIS. I think the significant and tougher part of my presentation is over. So, madam, any doubts you want to clarify now before I go to the next part of presentation?
Can we keep it at the end of the presentation?
Yeah.
So you can start writing the doubts in the chat box.
Fine. Okay, madam. Then, so from here, the presentation is hardly 10 minutes and simple. So entropy is another form of technology. So when you start a business, people will try to compete with you. So in the same way, when BIS is there, another company comes with another algorithm. And finally, it is also a kind of EEG derived parameter only, but the philosophy that they use for entropy is slightly different. So what they try to tell is, whenever patient is awake, the EEG is irregular. And when the patient is under anesthesia, the EEG becomes more regular. That is the concept they have chosen. So they try to find the irregularity in the EEG signal. And the more the irregularity, the higher number they give. And the more stable and regular it is, the lesser number they give. That is how the entropy scale has been designed by the algorithm. Entropy algorithm is out in the market, but this algorithm is not there. So in entropy, when I was using entropy in NIMANS, I actually liked it better than this for one reason. It gives two parameters, named state entropy and response entropy. So state entropy is the pure EEG signal. It doesn't have the EMG contamination because the frequency range of state entropy is 0.8 to 32 Hz, where the EMG starts from 35. So state entropy gives me a more accurate understanding of the anesthetic state. And response entropy is another number which entropy gives, where the frequency range is more broader. From 0.8 to 47 is the range which it covers. So here EMG artifact is there. So why they kept response entropy is, I'll tell you. So basically, if a state entropy number is around 25 and response entropy number is 35, the difference is around 10. So that difference is the EMG activity. So by looking at the difference between SC and RE, we'll come to know what is the EMG activity. So what to do at that time, I'll tell you. So this is how you place. It's simple to connect than this, only 3, 1, 2, and 3. 1 is a ground, 2 is reference, and 3 is the active electrode from where the recording is done. So here you can see the state entropy is 41, response entropy is 48. The difference is 7. This is a very good plan. Here the EMG activity is very minimal. So this is what I desire during the anesthetic management. If the gap increases more than 15 or 20, then EMG activity is significant. So that is the time you need to deal that. So studies are showing that RESC difference is directly correlating with the nociception more than muscle relaxant activity. So what they recommend is to give a bolus of fentanyl or morphine during that point of time, than giving a muscle relaxant, or increase the depth by using propofol. So this is what I covered. And another monitor which has come up, but not very famous, is called as Narcotrend. What they try to give is, they try to give alphabet along with the number. So since I don't think there is much novelty to the technology, that is why I don't think it has survived the test of the time. So E is where the patient is, A to E, you'll be given A is awake. E is where patient is in a good plane. So based on the alphabet, you can tell what is the depth of anesthesia of that particular patient. And there are studies comparing Narcotin and BIS, and it is almost comparable. And this is, I don't call it my favorite, but I'm finding it better than other monitors. It's called patient state index. So patient state index is also a kind of algorithm which gives a number from the EEG, but it gives a number not from single channel, it gives a number from deriving the value from 4 channels. So patient state index, if you can see, these are the 4 channels of EEG, and this is the number that you get. So this is how a patient state index sensor looks like. So it is attached like this bilaterally, and the advantage is right side and left side also you can differentiate. There are some surgeries in neuro where carotid surgery will be going on, where I'm interested to know where the clamping is done, because that is a side where ischemia can happen during clamping. And during ischemia, there are certain ways to identify the ischemia looking at the EEG. It is beyond the scope of this particular presentation, so let us not go into that. So that this monitor helps me in that area. So this is called as patient state index. The number is slightly different to BIS. So BIS is measured between 40 to 60 is called as ideal plane for BIS. For patient state index, it is between 25 to 50. So if a patient is between 25 to 50, then you call it a correct plane. It is one thing you need to know. So this is how patient state index electrode or sensor looks like, and they call it SEG line. Okay. This is a small video I took, patient state index. So you can see the 4-channel EEG working, and the number is 23. 23, I'm okay with that. As the neuroanesthetists, we try to keep the patient in slightly deeper plane since we don't use muscle relaxants. We are okay with that. And this also gives NIRS, that is NIRS beyond the scope of discussion. And cerebral state monitor was available. Now I don't think anybody uses. This is for freelance practicing anesthesiologists. It's a handheld device, and you can just carry it in your pocket and connect it, and it gives a number just like this. So I told you NICE guidelines and FDA, they don't recommend. I mean, they recommend, but they don't call BIS as absolute necessary. You may use it, or you might use it. That is how they recommend. So it is up to you whether you really want to use it or not. But I have an obligation to use. So it's not that if you use BIS, can we prevent awareness? There are some articles where they found that despite of using BIS, patient is aware. Maybe this kind of cases happen when you don't understand how the number is derived. When you don't understand how the waveform looks like. So when I see this kind of presentations, what I feel is you need to understand the waveform more than the number. So when you understand the waveform, which I explained in the initial part of the video, when there is a wavy pattern, when there is an alpha and delta, then by looking at the wave, you can make out. So maybe some particular cases, if I am there, I would have told that this is an artifact. Don't follow this particular BIS number. This is clearly an artifact. If either the electrode is loose or it's catching some kind of a ground electrical activity. So that is how a waveform is more important. So now presentation is over. Now let me tell you what happened in the cases which I initially explained in the beginning. The cervical ITM case where I didn't use the base anesthetic of propofol, where I kept the patient only on dexam and ketamine. Did the patient move? Yes, patient moved twice in the initial 1 hour. Then I had to go up with the depth of dexam and ketamine. After that, he didn't move. In the crucial stage of tumor resection, he did not move. Was he aware? Since the EEG showed that patient is lighter, And was he aware post-operative in the post-operative period? No, patient was not aware. Patient was in a very comfortable, and he told the experience is very good. And then third, neuro monitoring. Yes, the neuro monitoring, the motor evoked potentials came comparable to propofol. So I can't comment that it is superior because with one case, to do any comment, we need to have a series of cases or a study. So I got waves in that particular case. Post-op recovery. Post-op recovery, we ran dexam and ketamine for a significant time for 8 hours, but patient actually woke up in 15 to 20 minutes. So that itself shows that there is some, this anesthetic is actually good. And one unique thing what I observed is the patient hemodynamic stability was far superior than me running TCI propofol. I don't know, I can't attribute it directly to ketamine, calling it the sympathetic activity it would have maintained. But what I really think is the hemodynamic stability was very good despite of blood loss. Patient's hemodynamic stability was excellent. And one more thing is, at the end of the surgery, I thought I'll stop this and convert the patient desflurane for a faster recovery. That is the time when he got hypotension, not during the blood loss or during the surgery. So there is something unique with this anesthetic combination. We need to explore more, I think so. So these are the things which happened in this particular case. And another case which I showed you, where the patient, what the case was going, but the patient was a little in a lighter plane. So in that case, what we did is we had to increase the TCI and give a dose of morphine. And in spine surgery, I explained. So this particular case where despite of running 5.5 TCI and XM of around 13 ml per hour, that is like 0.7 to 1 mic per kg per hour, the patient was still in a lighter plane. So here we have to increase the anesthetics. So that is how the depth of anesthesia monitoring will help if we have a deeper understanding about it. So take-home points would be follow the trace, EEG trace, than the number. I explained you clearly why you have to follow the trace, and this is reliable only with a good signal quality index. If signal quality index is less than 80, don't rely on the number. And entropy can be helpful in identifying the nociception. So predominantly we'll discuss about this, but there's so many monitors coming up. But this is famous and popular because it is the first to enter anything. So that's it, guys. Thank you very much. That's all from myself on depth of anesthesia. Thank you, madam, for giving this opportunity.
Thank you so much. It was a pleasure. And you actually really cleared, I think, most of our doubts. Just a couple of questions before I get some from the participants. This can you tell us some more tips and tricks that we can use to decrease the impedance that we get in the theater?
So a good cleaning before attaching the bis, and good cleaning with sterilium or spirit, and then drying it up and connecting it. And after connecting, you have to secure it properly. I use IO-band for securing. So because all of the plasters will eventually come off during surgery. My case is, if it is a supine position or any other position like a beach chair, or those things are fine, but most of my cases are prone position, since predominantly spine surgery is where I use. So in prone position, first 1 to 2 hours, they work very well. Later on, slowly it starts coming off. So even despite of being putting IO-band, they come off sometimes. So that is why sometimes I get irritated with the S values. And nowadays I'm using only EEG for interpretation, since we have a neuro monitoring device in the operating room. And the EEG device comes with a corkscrew electrode, so their impedance doesn't matter because it is in the skull directly. There is no impedance question. So, but there is a significant problem. I think the company should address that in prone position. But in supine, usually 3 to 4 hours, nothing will happen. But with sweat, if the impedance is going up, maybe the signal quality will come down. I noticed something that if I was putting more of earthing plates to most of my monitors,
and to the table, I felt that the impedance was a little, the SQI would go up a bit. Do you think
Okay.
I'm correct, or is it just a...
No, what were you facing, madam?
The earthing plate, like earthing the cutlery machine, the table, the shock machine, and IDP machine.
Basically, earthing, I'll discuss about earthing now. So what we do is the neuro OTs are designed in a different way, madam. So at the time of design, biomedical engineers will sit with them. We'll explain something called as double earthing concept. All the electrical circuit, all the electrical plugs where you connect devices need to be double earth. So there is a procedure for that. So when you construct the OT only, it needs to be done. It can be done later on, but you have to stop the OT, which will not happen in any hospital. So during the construction time only, we tell the biomedical guys. So wherever neuro cases are possible to be taken, the plug should be double earth. When you do that, that significantly decreases the electrical artifacts or earthing artifacts. Other than that, we will have ground electrode. Ground electrode is there in the base. It tries to prevent, but since you're telling you're doing whatever techniques you're doing, actually in the double earthing, it will be sorted out. But if you don't have, those things will help. It is all about neurophysiology is more about artifact removal and everything, understanding where the artifacts are coming from. Even your table generates an artifact, light generates an artifact, the warming device generates an artifact, pottery generates an artifact, and every device which is being used in OT generates an artifact in the neurophysiology. Sometimes it is not possible to remove everything. That's why sometimes you need to accept the way with a small amount of an artifact. That time it's better to go with the waveform than the number. But double earthing will prevent most of these problems.
Another question is that ketamine Dexam, which you just showed us, which you used in your cervical case, what was the dosage that you used? So
ketamine, see, because here we're not using the base anesthetic. It's only one case I did till now. I don't have a significant experience, but that case, but ketamine regularly I use for all the scoliosis, deformity corrections, prospect guidelines recommend ketamine along with propofol. So I am used to using ketamine. It's not a new thing, but without propofol, giving ketamine is a tougher task. So there you need to escalate the dose. So 1 mg per kg per hour is the dose I give. It's a little higher dose, but what happens is if I was cutting down, patient was bucking. So I started with 0.5, we went up to 1. And Dexam started with 0.5, went to 1 mic per kg per hour. With this dosing, patient was not moving during the procedure.
Okay.
And a dose of fentanyl and morphine also needs to be given.
Okay.
Fentanyl at least needs to be given.
Madam, any comments, Dr. Naik?
Yes. It's really a pleasure to hear Dr. Masapu. Very nice. One of the clear, very lucid and very, very clear. One of the loveliest lecture I've heard.
Thank you, madam.
And you're from Bangalore. I'm also from Bangalore. I did marriage, changed everything, and I got settled in Mumbai.
Oh.
Most of my questions are answered, but still I have got few questions.
Okay.
This number one question is: the EEG corresponds to adult EEG by 5 years. So you can use this monitor.
Your signal is getting broken.
Madam, your voice is breaking a bit. Could you repeat the question once again? Yeah, the EEG pattern corresponds to adult EEG by 5 years. Can you hear me, Kalpana? Yes, madam, we can now. Yeah. So can we use this monitor below 1 year of age? Is it accurate?
No, according to recommendation, less than 6 months of age, we are not supposed to use, madam. FDA has not approved, but I'm trying to use in different age groups. You get a good waveform and a number. So based on the waveform, I can make out now. As I explained, if there is a alpha and delta waveform, I will be knowing the patient it is in a good depth. But with this very nice, beautiful wave will come. Actually, the impedance is lesser in children. You get a superior quality index waveform in children. Yeah. But FDA approval is there below 6 months of age.
See, and second is that BIS monitor. There are different monitors are patented around different algorithm. Like this monitor uses only EEG.
Yeah.
So it monitors the depth of hypnosis.
Yeah.
But do you prefer a monitor which also monitors the depth of analgesia, nociception?
Yes.
Along with it, because most of the time you are given extra dose of fentanyl, propofol to.
Correct.
So,
I
isn't
understood.
it?
I got your question. Basically, hypnosis and nociception, if you give me both monitors, I would use both. So ANI monitors have come up actually. Anti-nociceptive index monitors have come up. The 0 to 100 scale will be there. 100 is, it is opposite there. 100 is where the analgesia is like perfect. And 0 is where analgesia is inadequate. We had to keep the patient between 80 to 100. That is one. And GE company guys also something called surgical pleth index. So even there, they give a square box kind of a thing where the dot actually moves from one box to another box in a way. So that is also,
I've used it. Yes,
yeah, that is also one way. And all of these are derived from this and this one. If you actually have the advanced technology, it's always better to use. So, but it's a personal choice.
Yeah.
Surgical pleth index I have used, but I have taken review from many of my neuroanesthesia colleagues. They were not very happy with that. It is good to tell that it's a surgical pleth index and tells you everything. How much information it is giving, how much value it is adding is questionable, is what they felt. But many people actually use that. ANI monitor also was same. So nociception should be monitored or not, because what happens is if you give a dose of fentanyl, and most of the times the ANI monitor gives 100 value for a significant duration of time. But it is better to use. But some people might think it's an overkill. Having so many monitors.
But no, that is the depth of awareness as well as analgesia would have been a better monitor. I just want to know why do you prefer which depth of anesthesia monitor? Why you don't like? Why you don't like me? What is the disadvantage of using entropy?
No, entropy, there is no major disadvantage, madam. Actually, in order, if you want me to place.
Even narco trend is, I felt it was superior to this. This is absolutely basic monitoring.
Basic, yeah. So, that's what in order, I would keep, number one, I would keep SEDline as my recommendation. Number one. Number two would be entropy. Number three will be BIS. But BIS has so many fans in the world, actually. They might oppose us, but because many people, they got used to BIS, so they don't want to change now. So they might oppose what I'm telling. But SEDline is the one I'm feeling it is better because I'm telling it's a clear
Massimo's.
Massimo, yeah. 4 channels it is taking.
It's good.
Bilateral it is taking. So patient station, I'm feeling superior. Even though my experience is only 2 weeks, I can't come to conclusion. BIS I've been using from years actually. So, but I feel better.
You said the BIS monitor is not of much value in certain cases in neurosurgery, like TBI or even you said about the tumors also. So, is there any comparison between the BIS value and GCS?
Yeah, I did a study, madam, and GCS and BIS are directly correlating inversely. So whenever the BIS is whenever the GCS is low, BIS is also very low.
Very low. Yes.
Yeah. So
there is no use of using this in traumatic brain injury or brain tumor cases. In the frontal lobe tumors or.
No, what happens is during the surgery when they raise the flap, the contact will go, madam. Yeah.
That's what I feel. That's a question I wanted to ask you. How do you use these electrodes? No matter you use IO band or whatever, ultimately there is a shift in the.
I don't use in craniotomy, and surgeons are not allowing also in the field if you are putting nowadays. So I predominantly use BIS in spine surgeries, because I'm worried about overdosing the spine patients, and I had to get neuro monitoring. So the depth should be very perfect. I mean, good range of depth. Too much deep, if I give the patient, neuro monitoring won't come. The MEP and SSCP won't come. Too much light, if I keep, patient will buck. So that is where I found the problem, and this is useful. So most of the medical science, what my personal opinion is, if a problem is there, we have to solve. Yeah. So that is where I found a problem. And in brain surgery, I don't have any major problem with depth of anesthesia. So that I don't think we have to solve that problem. But that's my opinion.
Yeah. Just next, one more question is that history of epilepsy.
Yeah.
Or there is any neurological disorder which has affected the EEG preoperatively.
Yeah.
So is it better to have a reference EEG preoperatively, or you think that makes any difference?
It makes a difference. Before induction, if you connect the BIS or entropy or whatever monitor and take a baseline, their baseline might be different, because a patient of Parkinson's is coming for a surgery, and they will be a little dull, and their baseline BIS must be 75 or 80. There you cannot target. For them, you need to understand that their baseline itself is low. So you maintain a range between 40 to 60, but understanding the baseline helps in how much induction agent to be used, how much maintenance agent to be used. So that will be very helpful. Epilepsy is a different thing. Epilepsy, if the patient is on multiple anti-epileptics, like refractory epilepsy patients, if you observe, the cognition will be slightly lower. Their baseline BIS will be low. So even then, you might have to take a baseline. But practically, most of the time, this is connected once the induction is done. But it is always, if you have to talk in an ideal way, like an idealist, if I have to talk, then better take it before induction, so that you can titrate your induction. You can titrate the
yes. Thank you very much, Dr. Dheeraj. It was really nice listening to your talk. We really enjoyed it.
Thank you. Thank you.
Kalpana, can I ask one question?
Yes, Dr. Dheeraj, this is Dr. Dhaigude, madam. She's one of the very senior most pediatric anesthesiologists in Bombay. And madam has been kind to join us all the time for this clinical series. And madam, please go ahead.
Yeah.
Thanks for joining, madam.
Awesome. I have two questions. What about the patients with cerebral palsy? If they come for spine surgery, what would you find? Will that be appropriate monitoring you will get with BIS or narcotrend or whatever?
No, same thing, madam. Cerebral palsy also comes in the category of neurological disorder, where the baseline itself will be low when you connect the BIS. So there, it's very useful. If a cerebral palsy patient is coming for a spine surgery, and then you connect BIS in that particular patient, you can titrate the maintenance maybe with my knowledge, with 2 to 2.2 of TCI, also you can actually get a very good depth. Or if you're using inhalation, with a MAC of 0.3 to 0.5, in that range itself, you can actually get a desired anesthetic depth in these patients. So you'll end up not overdosing them. So in this
actually, we do assume that they need almost more than 10% less dosage. All these cerebral palsy patients. So another thing I wanted to ask, is there a marked difference in EEG pattern in patients with in the age group of 20s and age group of 70s?
So EEG wave is same, madam, in any age group from birth to death. EEG pattern and EEG waves are almost similar. To my knowledge, neurology is very complex, but the thing what differs is in an age group of 20s, the sensitivity to anesthetics is they're very resistant, I feel. They're not resistant, actually. They're not very sensitive. So you have to use higher anesthetics, higher range of TCI to get your desired range of BIS. In an older age, you use a lesser dose to get desired range. But waveform per se, I don't think there is any difference, because I constantly see the EEG every day, actually. Hours and hours of EEG I'll be looking at. And I don't see a change in the wave pattern and structure and the morphology of wave. Only thing which matters is with what concentration of anesthetics, how the depth that you're achieving matters.
To get the proper depth.
Yeah,
okay.
The depth matters. In young people, actually, up to 5.5 TCI, you might have to use if you're not using any adjuvants like dexmedetomidine. And if you're using dexmedetomidine, maybe a TCI of 4 to 4.5 will be required. In older people, usually a TCI of 2.8, 2.5, sometimes 2.2 TCI also will be able to get the patient under control without muscle relaxants.
Very good. Yeah. I mean, in pediatric anesthesia, we hardly get chance to use this base monitoring.
Yeah.
Or,
Pediatric
general
concerns
cases.
are different now.
Because of the growing
I will
brain
use
and
I have to use for pediatric also, because most of our pediatric cases are meningomyelocele, tattooed cord releases, and brain medulloblastomas in the posterior fossa. Every case requires neuromonitoring. So to understand, while doing neuromonitoring, we catch EEG also from the and we monitor them.
Very good. Very nice presentation, I must say. You made everything very clear.
Thanks, madam.
Concepts were made very clear. We had just read it from the books, but I think you explained everything very nicely.
Yes, this topic is very tough actually. For me also to understand, it took many years.
Very complex subject.
Yes, madam.
Dr. Dheeraj, there is a question in the chat box. Dr. Saji has said that, thank you, sir. This has been simplified, but he has two doubts. First one is ketamine in neuroanesthesia case with raised ICP. Is it okay? Any dose to limit or only with dexmed and propofol combo? Number two, in a resource limited setting with only ordinary infusion pumps and no BIS, what's your earlier technique or advice to achieve smoothness, intra-op and extubation on table? Is it possible? Example, for a long decompressive craniotomy, as we mostly ventilate them overnight and extubate them next day. So his first question is, a rationale of using ketamine in a neuro.
Yeah, ketamine is a drug which is being extensive research nowadays. So the older concept is that ketamine will cause glutamate excitotoxicity, and it raises the intracranial pressure. That was what was thought. But see, when you're, for example, I'll give you one or two scenarios. You're inducing a head injury patient. Okay, where you're really concerned about ICP is head injury patients or very big brain tumor with significant perilesional edema and a mass effect. These are the situations where you're really worried about ICP. So when you're inducing those patients, you're actually happy to use propofol. You can induce it propofol, which has cerebral vasoconstriction effect. It decreases the ICP. It also has a neuroprotective action. So there is, I don't feel any major reason to use ketamine there. If you think that ketamine can maintain BP well than propofol, that is the older concept that is gone now. You have to take an arterial line, start an RF during induction or phenylephrine during induction. So I don't see the role of ketamine during ICP situations. Ketamine has a role. The research is being done in epilepsy. In epilepsy patients, when we manage in neurointensive care, I have done a study also on that. And we run ketamine for refractory cases. That is where majority of research is going on. And ketamine usage in spine surgery. Prospect guidelines has given as grade 1 evidence. Ketamine, they are recommending for all complex long segment fusions and deformity corrections. You have to run propofol and ketamine also to be run 0.25 to 0.5 mg per kg per hour. These are the two areas where ketamine is being used by us and not in the raised ICP. I think that will answer the first question. And the second thing, without TCI, see, TCI has come into the market actually from maybe 4 to 5 years. And actually it's being practiced from 1 to 2 years. I've been using TCI from maybe 2 to 3 years. Before that, I was doing neuro cases from last decade. So we were using propofol. And I actually how we do is Bristol regimen is there. So 10, 8, and 6. So we have to initially run for the first 10 minutes for 10 mg per kg per hour, next 10 minutes for 8 mg per kg per hour. And after that, you have to continue the patient for 6 mg per kg per hour. And to achieve a target of 3 micrograms per ml of TCI level. So you can follow that. And 2% propofol I used to use before. So, and we used to run in a range of around 20 to 25 ml per hour, 2% to calm the patient without TCI propofol in a male patient. In females, I used to use around 18 to 20 ml per hour of 2%. So that was how I was doing. After shifted to TCI, I didn't have, now I don't have to do all the calculations. And about ventilation is third question. Ventilation, if we are planning to do ventilation and for a head injury patient, there are two things important here. After the head injury, most of the surgeons want to assess the GCS of the patient. They will ask you to reverse the patient once. So what we do is we shift the patient to ICU and then decrease the dose of TCI to around 0.5 to 1. And it will just keep the patient calm, but not sedated. The surgeons will assess the GCS once. And once they're comfortable, then we'll resedate them for neuroprotective measures. And if the patient is not recovering after decreasing the TCI to 0.5, then it is not sedation. There is something wrong in the brain. We have to take a CT scan and see if the hemorrhage is expanding inside or if some other problem is there. Based on that, the next steps will be done. So that is how we have to ventilate. Ventilating post procedure is a complex thing, actually. Many people think it is simple. There, you have to spend more time there than in OT.
Dr. Adira, just a addendum to the same question. When you're telling us to use the Bristol regime of 10, 8, 6, 1, 1 hour, and then the 6, 1 can continue for 4 to 6 hours, that may cover your surgery for, say, 7 to 8 hours, right? I mean, 6, 7, 8, 9 hours. What happens if the surgery goes a little longer? Do you continue?
You have to decrease the dose.
Decrease it again by... Okay. There's another question which says that, is there any post-op advantage of using this? I think you just showed one slide, but she says, I read some article that it helps in decreasing post-op cognitive dysfunction. And if yes, how does it do that? That's what Dr. Bhavya would like to know.
Post-operative, I have not used this much. So if the patient is drowsy or delirious, it makes sense to use this there to understand what depth they are in. And you can understand the trend in which they're coming out. For example, last half an hour before, the patient is having a BIS of around 65 to 70. And the patient is delirious. Now the BIS is around 80. And patient is calming up a little bit. Then the trend is more towards recovery. So post-op might help, but it is very tough because what happens is when patient starts moving, it is this catches the artifact. But if patient is calm, then it'll help me. But I don't usually use in the post-operative period. Once extubation is done, I'll remove everything and then I'll shift the patient out. And most of the patients we extubate now 50% we extubate in OT, 50% in ICU. So what we do is, but either way, I'll remove and send to ICU. If you're extubating in OT, I'll keep the BIS till then. But post-operative, I don't have much experience.
One of the videos you showed that you also had a Dexam infusion on with a pump.
Yeah. Yes, propofol and Dexam is what I use.
Yeah, which model do you use for the Dexam?
Dexam, I don't use a TCI model. I just use manual calculating.
Manual, okay.
Manual. I don't. The new TCI for Dexam is an Eleveld, I think. You have experience.
Eleveld, even Hanabat port was earlier used. I don't know. If you have any practical experience. Yeah, okay.
We use Alaris pumps where we only for propofol 1%, 2% we have models in ETS, Kataria, and Schneider.
Correct.
These three things we have.
Okay. Are there any questions? Any more from anybody? If not, then Dr. Dheeraj, I take this opportunity to thank you so much for spending this Sunday morning with us and making this topic so interesting, so lucid. And I think all our students are going to go back with more confidence in using this and understanding it better, I hope, from tomorrow.
Thank you very much, madam. Actually, thanks for giving the opportunity. I never presented in this forum until now. And hope we'll do more collaboration.
Of course, we will. Yeah. Thank you so much. And I thank everybody for joining us this Sunday morning. Thank you.
Thank you. Thank you.
Bye.
Thank you very much.